8:30. So we have our presenter, unfortunately not in person because he's frozen in Minnesota to the ground, but we welcome him very warmly to tell us about his research. And we'll have a perfect introduction here.
Hello and good morning to everyone in person and on Zoom. For those of you who do not know me, my name is Jacob Diaz, and I am a medical student researcher in the Andrew Brown Lab. It is my privilege today to introduce Dr. Glenn Lobo, a distinguished researcher and educator whose work has profoundly advanced our understanding of vitamin A metabolism. Dr. Lobo is Associate Professor of Ophthalmology and a faculty member in Neurosciences at the University of Minnesota. With over 12 years of experience in genetic, molecular, and cellular biology, he has made a significant contribution to the field of visual neuroscience, particularly in studying the mechanisms that support vision. Dr. Lobo earned his PhD in human genetics, which laid the foundation for his career in vision research. During his postdoctoral training in Case Western Reserve, he worked under Dr. Johannes von Lintig, where he identified key pathways involved in vitamin A uptake and metabolism, a nutrient critical for vision function. His work during this time provided insight into the absorption, storage, and transport of vitamin A in the liver and the eye and the body, studying its essential roles in chromophore production and phototransduction. He continued his research in the Cleveland Clinic's Cole Eye Institute, where he established zebrafish mutant models using cutting-edge gene editing techniques like TALENs, where he discovered novel mechanisms for retinal ciliopathies, including the role of endoplasmic reticulum stress and TULP1 knockout mice, a model for Leber congenital amaurosis. Since starting his own lab at the University of South Carolina in 2016, Dr. Lobo is focused on investigating the molecular and physiological roles of membrane receptors like RBPR2 and vitamin A transport. His innovative work using zebrafish and mouse models has shed light on how these receptors regulate the delivery of vitamin A to the retina and maintains homeostasis under variable dietary conditions. His research has also revealed how disruptions in these pathways can contribute to the retinal diseases such as retinitis pigmentosa. Supported by multiple R1 NIH, Knights Templar, and Vitreoretinal Surgery Foundation grants and awards, including the prestigious DCI New Investigator Award, Dr. Lobo and his team are advancing our understanding of vitamin A homeostasis and its relationship to visual signaling and retinal health. In addition to his research, Dr. Lobo is an active member of the academic community. He serves on advisory and admissions committees, chaired a research subcommittee, and has a position on the University of Minnesota Senate and holds associate editor positions in leading journals in ophthalmology and cell biology. His commitment to mentorship and collaboration reflects his dedication to advancing the field and inspiring the next generation of scientists. It is an honor to welcome Dr. Lobo on Zoom today. And please join me in giving him a warm welcome as he discusses the physiological role of vitamin A receptor RBPR2 in the whole body retinoid homeostasis.
Thank you very much for that kind introduction, and I hope everybody can hear me well. I would also like to thank Dr. Palczewski and Dr. Palczewska for the kind invitation, and apologize for not being there in person, because given Minnesota, we never know the weather. So, flights in and out could be a problem. So let's get started about vitamin A receptors in whole body retinoid homeostasis. So the way the talk is going to be set out is going to be in this format. So I'm going to give you an overview of background information and historic perspective on the biological function of dietary vitamin A metabolites, cumulative called retinoids in human physiology. The second part of my talk, I will introduce two vitamin A membrane receptors, STRA6 in the eye and RBPR2, which is expressed in non-ocular tissues. I will further do a deep dive into the diverse tissue expression patterns of these two vitamin A membrane receptors. And then finally, I will present to you unpublished data which establishes a synergistic role of the diet, vitamin A, and genetics in maintaining whole body vitamin A or retinoid homeostasis. So what is the definition of a vitamin? So according to the World Health Organization, the requirements for a vitamin should be the following. It should be a micronutrient. It has to be a dietary component. It has to be essential or vital. In other words, not produced in the body, and has to be obtained from the diet, from food. And most importantly, it has to perform some physiologically important functions in human. So this brings us to the question, is vitamin A an essential nutrient with biologically relevant function? So humans cannot produce vitamin A de novo in the body. So we have to obtain it from two different sources. The first one is from pre-formed vitamin A or retinol, and we get that from liver, meat, milk, cheese, which is basically animal sources. And the second source is from provitamin A carotenoids, such as beta carotene, which is found in leafy vegetables and fruits. So although these preformed vitamin A and plant-derived foods are inert, they have to be converted into biologically active form of vitamin A called retinoids. And I won't discuss in detail in this slide, but in the following slides, I'll discuss in detail the absorption and the metabolism and the transport mechanisms of these vitamin A products called retinoids. But vitamin from animal-derived sources or plant-derived foods all form into retinol or retinaldehyde. They're then converted by LRAT into retinal esters, and they're basically stored in the liver. And depending on the body's requirement for vitamin A, they're transported bound to a protein called RBP4, which enters target cells. In the target cells is further broken down to retinal or retinoic acid. And as we know that retinoic acid is a potent transcription factors, it binds to these RAR elements on gene promoters to effectively initiate its transcription. So just an overview of different forms of vitamin A. Retinol is the major transport form of vitamin A in the serum. Retinal esters is the main storage form of vitamin A in the liver and other tissues, including the eye. And retinoic acid, then another biological active form of vitamin A, is involved in gene transcription. And as we all know, 11-cis retinaldehyde is very important for visual function. So vitamin A and its metabolized retinoids play a very important part in embryogenesis. It is important for tissue differentiation, hematopoiesis, and for organogenesis. It's also been showed in zebrafish models that it's very important for patterning of the retina. And in children and adults, vitamin A is important for vision, immunity, and functioning of the human brain, shown in the figures on the left. So with the exception of 11-cis retinaldehyde, which is important for visual function, where it binds to the opsin protein in the retina to form rhodopsin, the remaining biological function of vitamin A has been attributed to its transcription regulated retinoic acid. So even given the importance of vitamin A for decades, we know it's very important for health, metabolism, visual function. Vitamin A deficiency still remains a global health concern. And shown here in the global map, especially countries from Africa, Southeast Asia, even the Pacific, and some countries in South America have large populations, almost 50 to 60%, up to 80% of their populations suffer from some sort of vitamin A deficiency, which can lead to conditions such as night blindness, which is caused by decreased rhodopsin production, dry eyes, weak immune system, delayed growth, metabolic syndrome or increased adiposity, infertility. And if these conditions are not treated earlier on, they can lead to much more severe conditions such as increased mortality in children and eventually blindness. So what are the various routes of vitamin A deficiency? So shown here in this figure, there can be a few routes that can happen through the diet or through genetics. So obviously the first route is deficient intake or low intake of dietary vitamin A rich foods. It can also happen through genetics, where genes or proteins involved in its storage absorption are defective in human beings. Both these conditions can lead to seriously low amounts of serum retinol or serum vitamin A in the blood, and eventually lead to eye pathologies such as night blindness and then photoreceptor cell degeneration. So given this, we believe that both the diet and genetics play a very important role in maintaining vitamin A homeostasis in human beings. So we are interested in studying the mechanisms that regulate vitamin A absorption, storage, and distribution to the eye and other tissues in the body. And this is very important to prevent these conditions in humans. So let's dive deep into these three major aspects of vitamin A. We'll talk about absorption, storage, and distribution of dietary vitamin A. So I know it's a busy slide, so just bear with me. So as mentioned earlier, human beings cannot produce vitamin A in the body. So they have to acquire from provitamin A carotenoids, this is beta carotene, or preform vitamin A. And beta carotene has been shown by the one-linked lab to be actively absorbed through SRB1, while retinol can diffuse into the intestines of the erythrocytes. From there, they are packed into these chylomicrons as retinal esters. They're all converted to retinal esters and packed into chylomicrons, enters the circulation, and then goes into the liver, which is the major vitamin A or retinal ester storage hub. So in the liver, the chylomicrons enter as retinal esters, and they can be converted into retinol. And depending on the body's requirement for vitamin A or retinol binds to this protein called as RBP4, the retinol binding protein 4, and then is again excreted out into the circulation and then distributed into peripheral tissues, including the eye. In the eye, there is a receptor called STRA6 present at the basolateral surface of the RPE cells. And this receptor for vitamin A is important for binding to RBP4, and that allows retinol to enter into the RP and then into the photoreceptors to bind to opsins. So the major receptor for serum RBP4 bound retinol in the RPE or the eye is STRA6. And then accordingly, it has been shown since 2007 that mutations in the STRA6 gene associated with this Matthew-Wood syndrome, it combines a various amount of pathological conditions throughout the body. And the main ones being ocular related is microphthalmia, where you have smaller eyes in humans or in the babies born, or anophthalmia, where they are born without eyes. And this has been attributed to lower vitamin A in the eyes. So lower retinoids in the eye can lead to these congenital conditions. So what are the memory receptors identified so far for binding RBP4 for intaking of retinol into the system? So various groups, including Sunich from UCLA and Johannes von Lintig's lab in Case Western, have shown that STRA6 is the major vitamin A receptor in the eye. However, STRA6 is not expressed in key tissues such as the intestine and the liver, which raises the question, are there other receptors for RBP4 retinol in these organs? And then first identified by Graham's group in the University of Utah, they published a paper way back in 2013 showing that the receptor for RBP4 retinol in the intestine could be the second receptor, RBPR2, called the retinol binding protein receptor 2. So this is the screenshot from that publication in JBC, where they identified a second retinol binding protein for vitamin A in the liver. And using in vitro analysis, they confirmed that RBPR2 is a novel RBP4 receptor that can mediate retinol uptake into tissues, which is present in systemic or peripheral tissues. So then our question was, what are the tissue expression patterns of RBPR2 and STRA6? Are they the same or do they differ? So this is an early publications from the Von Lintig and Palczewski lab in cell metabolism in 2008, using zebrafish model and using whole mount in situ hybridization, where they found that STRA6 was expressed in the pineal glands and then in the eyes of 4-day-old zebrafish, and specifically in the RPE cells of this. And we can see at 24 hours, there's no much of expression in other tissues of the zebrafish, indicating that this could be very specific to the eye. In our analysis for RBPR2, the second receptor, we found that RBPR2 has distinct patterns to STRA6, right from 3 days up to 5 days post-fertilization. We found RBPR2 expression to be highly expressed in the liver, intestine, pancreas, and spleen, and not in the eye. And then cross-sectioning it here, showing you an F, G, and H panels, we find it to be expressed in the liver, intestines, and pancreas, but not in the eye. So straight off the bat in zebrafish, you can see that STRA6 and RBPR2, two receptors for vitamin A, have very distinct patterns in this vertebrate. Let me ask the question, what is the tissue expression patterns of RBPR2 and STRA6 in mice? So we first performed semi-quantitative PCR, and we isolated eye, heart, and various peripheral tissues from mice, and conducted a semi-quantitative PCR for both RBPR2 and STRA6. And as you can see, STRA6 is highly expressed in the eye, in the kidney, and to a lesser extent in other peripheral tissues, but not in the liver. And this is in contrast to RBPR2, the second receptor, which is expressed in peripheral tissues, but not in the eye. And we confirmed this by qRT-PCR, which showed a similar pattern that STRA6 is highly expressed in the eye, while RBPR2 is highly expressed in the liver, intestine, and other peripheral tissues, but not in the eye. So taken together, showing you a schematic model of mice and zebrafish, you can see from this that STRA6 and RBPR2 have very diverse expression patterns, with STRA6 shown on the top panel here in the mouse and zebrafish being highly expressed in the RPE cells or in the eye and the brain, and to a lesser extent in the kidney. And same goes for the zebrafish. On the other hand, RBPR2 is not expressed in the RPE or the eye, but is highly expressed in peripheral tissues, including liver, intestine, spleen, the adipose, indicating that all of these two might function the same way. They have very distinct and diverse expression patterns. So having said that, what is the proposed role for the second vitamin A receptor in whole body retinoid homeostasis? So it is believed that RBPR2 being expressed in the liver could be involved in the reabsorption of retinol from the serum in cases when there is a lot of retinol floating around in the serum, and being expressed in various peripheral tissues such as spleen, kidney, and lungs. It's also involved in the uptake of retinol from RBP4 bound retinol in the circulation to maintain whole body retinoid homeostasis. And this is quite in distinction to STRA6, which is highly expressed in the eye. So the proposed role of RBPR2 is supposed to be in whole body retinoid homeostasis as compared to STRA6, which is involved in eye physiology for vitamin A uptake. So we asked the question, is RBPR2 a bona fide RBP4 receptor? So to be a receptor, it has to physically bind to that. So 3 labs, including us, have proposed that RBPR2 and STRA6 contain extracellular RBP4 binding residues. And doing a homology alignment of these protein sequences from mouse, human, and zebrafish, RBPR2 and STRA6, we find there are 3 amino acids, SYL, that are quite preserved among these species. And compared to STRA6, there seems to be a common motif, although a little bit divergent. It seems to also contain those amino acid sequences that could putatively bind to floating or circulatory RBP4, and that mechanism could internalize retinol into cells. And then using computer homology modeling, we found that mouse RBPR2, the extracellular binding sequence is in very close proximity to RBP4, which further indicated that RBPR2 could physically bind to exogenous RBP4, and this mechanism is important for sequestering retinol into cells. So the proposed model for STRA6 and RBPR2 binding to its ligand is as below. STRA6 seems to contain similar binding residues like RBPR2. Unlike that, it's a little bit divergent, but it's still in that same motif. And then STRA6, the extracellular domain of STRA6 has these motifs like RBPR2, which can bind to circulatory RBP4. And that's the mechanism for internalization of retinol into cells. So the function of both these receptors are supposed to be similar, but they have divergent RBP4 binding residues on their extracellular domains. So then we decided to test this hypothesis in vitro. So for that, we generated 3 RBPR2, RBP4 binding domain mutants. And I'll go through that experiment. So we took mouse RBPR2 cDNA, the full length, and noted here as wild-type RBPR2. And then using site-directed mutagenesis, we generate individual SYL domain mutations in RBPR2. Clone them into a pcDNA 3.1 V5 tagged vector to finally get these constructs that are tagged with V5. And then we next determine the functionality of this proposed RBP4 binding domains on RBPR2 in vitro. So we took NIH TT3 mouse fibroblastic cells and we overexpressed the wild type RBPR2 and the 3 mutants. And as you can see here in green, and from the V5 tagged with the 488 dye, you can see for a membrane receptor, it nicely localizes to the membrane of these cells. In a similar fashion, all the 3 mutants also are localized to the membrane of these TT3 cells. We then confirmed equal expression of the mutants with wild type by Western blotting. And shown here, you can see that the mutants are pretty much expressed in the same quantity as the wild type in these overexpressed cells. And then doing a fractionation assay where we isolated the membrane fraction and cytoplasmic fraction of these cells, you can see the majority of the mutants, like the wild type RBPR2, localized to the membrane, with a small amount of 2 mutants localizing to the cytoplasmic fraction shown here. So overall, the take-home message from this experiment was that the mutants like wild type can localize to the membrane and are not defective in transport. So then we asked the question, is there any defective binding of RBPR2 to circulatory RBP4 retinol? So we acquired stable TT3 LRAD expressing cells from Dr. Wonlintig and Martin Golchak at Case Western. And then we overexpressed wild type and the 3 mutant RBPR2 plasmids in these cells to make stable cell lines. We applied exogenous retinol-bound RBP4, which was properly folded. After 24 hours of incubation of RBP4-bound retinol, cells were washed and lysed, and then subjected to immunoprecipitation and Western blotting. And then shown down here, you can see there's very strong binding of wild-type RBPR2 to its ligand RBP4 protein. But on the other hand, you can see defective binding or less binding of these mutants to exogenous protein, indicating that some of these residues can interfere with the binding of exogenous RBP4. And shown here is p-values, which is just a standardized testing of these RBP4 binding percentages over input, with RBP4 wild type constructs set at 100. And you can see about a 60% decrease in the binding efficiency of these mutants to circulatory RBP4, indicating that these residues on RBPR2 are important to bind to circulatory RBP4 for internalizing retinol. So the next question we ask is, do any of these mutants show defective retinol uptake from circulatory RBP4? So we took the same cells that were stable, expressing LRAT and individual wild-type RBPR2, or the 3 mutants. And in this case, we added tritium retinol bound to RBP4, incubated those cells for 60 minutes. Then cells were washed and lysed, and then we quantified tritium retinol using a scintillation counter. And this showed that unlike the wild-type RBPR2, which could take up retinol bound to RBP4 very effectively, the 3 mutants had defective uptake, as shown by the retinol content inside the cells. And they all varied in the amounts of retinol they could take up, but the overall message was they were defective as they were mutated in the interacting domain of these binding domains. So to summarize this part of the talk, there are 2 membrane proteins that exist for the uptake of circular retinol-bound RBP4. STRA6, which is highly expressed in the RPE of the eye, and RBPR2, which is highly expressed in the liver and non-ocular tissues. And showing you earlier in vitro assays, we confirmed that RBPR2 contains these extracellular RBP4 binding domains or binding residues. And these binding residues on RBPR2 are important for not only its binding to extracellular RBP4, it's also important for role internalization. So what is the physiological role of RBPR2 in whole body vitamin A homeostasis? So to answer this question, we generated a whole body RBPR2 knockout. To do this, we obtained cryopreserved conditional RBPR2 embryos from UC Davis Comp repository. Shown here is the RBPR2 gene targeting constructs with the LOXP sites. And after flip recombinase, we generate a RBPR2 conditional allele, and then recombinase by mating these mice with actin-Cre, we generate a whole body RBPR2 knockout mouse. So we first confirmed the generation of a whole body RBPR2 knockout mouse line by genotyping and Western blotting. Shown here on the left is a PCR for flox and wild-type alleles. And in the red box, you can see that flox and flox mice with the Cre allele here confirmed that we have a global knockout mouse compared to the other ones that showed flox wild-type with or without the Cre. And then we isolated from wild-type and from these mice, intestine, liver, and other peripheral tissues. While the wild-type, we saw strong expression of RBPR2. The tissues, intestine, liver, kidney, and lung in these knockout mice did not show any expression of RBPR2, indicating that these mice were devoid of RBPR2 protein expression. So we confirmed that we do have in hand a whole body RBPR2 knockout mouse line. So then we conducted a longitudinal study where we split these mice, wild-type and RBPR2 mice, into 2 groups. Group 1 was a start-sum study for 3 months, and Group 2 was a long-term study for 6 months. So these mice were fed either with a vitamin A sufficient diet of 4 international units per gram, or with a vitamin A deficient diet of 0.22 international units per gram. And this was done for 3 months and 6 months. So at the first instance, we were interested in quantifying the vitamin A metabolites in the liver, where RBPR2 is highly expressed. So shown on the top panels are HPLC traces for retinal esters and all-trans retinol. And on the right side, we have the absorption spectra of retinal palmitate and all-trans retinol. And as you can see in the panels before, looking at retinal esters in the wild-type mice on both different diets, they presumably can maintain retinal ester concentrations under both diets, while the RBPR2 knockout mice on the VAD or the vitamin A deficient diets cannot maintain retinal esters or retinol concentrations effectively. We do not, at this time point, we do not see any differences in the VAS or the vitamin A sufficient RBPR2 knockout mice. And looking at total retinoids, which is a cumulative assessment of both esters and retinol and other retinoids in the liver, wild type mice like the wild type, like the RBPR2 VAS mice can maintain total retinoids in the liver as compared to RBPR2 knockout mice on VAD diets. So this scenario changed drastically at the 6 months time point. Shown here are HPLC traces for the 2 vitamin A metabolites we analyzed. And you can see that while the wild type mice on the both diets can maintain total retinoid content in the liver, we find that now at this time point, RBPR2 mice have severe depleted retinal esters and retinol in the liver at this time point. So what about peripheral tissues where RBPR2 is expressed? So we took spleen, lung, and kidney and performed the same HPLC analysis where we quantify retinal esters and all-trans retinol in these tissues. And again, we observed that wild-type mice under both vitamin A supplementary conditions can maintain total retinoids in all these tissues. And this is in stark contrast to RBPR2 knockout mice, especially those on the vitamin A deficient diet that are unable to maintain total retinoid homeostasis in these peripheral tissues. So then we measured serum retinol binding protein in RBPR2 mice. And quite interestingly, we found that while wild-type mice can maintain serum RBP4 homeostasis under both vitamin A supplementary conditions, the knockout mice seem to have higher amounts of RBP4 in the serum. And quantifically, we saw here they were about 2 to 3-fold higher in these mice, which is quite surprising to us. It could indicate that this could be apoprotein, RBP4 in the serum. In contrast to wild-type mice that can maintain retinol levels quite easily in the serum, RBPR2 mice are unable to maintain serum retinol concentrations. And then quantification of RBP4 and serum retinol, looking at the ratio, we find that there's much more serum apo-RBP4 in these knockout mice under both dietary conditions, indicating that most of the circulatory RBP4 in these mice is devoid of retinol. So then we asked, what are the consequences of decreased serum retinol on ocular retinoids? The left panel, we can see the HPLC traces for ocular retinoids and the absorption spectra shown here. And here is a total retinoid quantification of retinol, retinal esters, and Levin's retinol in the eyes of these mice. So again, like wild-type mice are able to maintain total retinoid concentrations in the eye, even though they are subject to different vitamin A diets, the knockout mice are not able to do that, as shown here as decreased retinoid concentrations in the eye of RBPR2 knockout mice. And then what are the consequences of this on visual function? So we performed scotopic ERGs in vitamin A deficient mice and both genotypes, the wild type and RBPR2 knockout. We do not see a significant reduction in scotopic ERGs in the RBPR2 knockout mice on the vitamin A sufficient diet as compared to the RBPR2 knockout mice on the VAD diets, which means retinoid homeostasis can be maintained in these mice when they are on vitamin A sufficient diets. This picture changed at about 6 months' time. We saw that total retinoids in the RBPR2 mice are now more significantly decreased at the 6 months time point compared to the vitamin A sufficient or deficient mice under the wild type background. And this is reflected in the A-wave analysis shown here by scotopic ERGs and showing down here, there's a significant differences in scotopic ERGs at 6 months when you compared wild-type mice to RBPR2 knockout. So overall, the serum retinol decrease has a significant effect on ocular retinoids and then ERG function and visual function in RBPR2 knockout mice, indicating that rhodopsin, most likely rhodopsin quantities are decreased in these mice. So then using a protocol from Beata at Case Western Reserve University, we took eye globes from wild type and knockout mice, and using a 1D4 pull down, we estimated rhodopsin concentration by UV spectrophotometry. And in dark adapted RBPR2 mice, we saw a significant decrease in rhodopsin concentrations shown here, where you measure Levin's retinal at 500 nanometers. The opsin protein is at 280. And then when you compare wild type to RBPR2 mice on the VAS diet, there's not a big difference. But when you compare wild type to RBPR2 knockout mice on the vitamin A deficient diet, there's a significant drop in rhodopsin concentrations in the eye of these mice, indicating that long-term loss of RBPR2 can affect not only peripheral tissue retinoid homeostasis, including serum retinol concentrations, but also has an effect on rhodopsin concentrations in the eye. So what about the presence of mislocalized rhodopsins if rhodopsin is low? So looking at wild type mice on VAS or VAD diets, we can see there's not much of apoprotein opsins in the retinas of these mice, where rhodopsin is shown here in green, and then DAPI or the nuclei is shown in blue. But that scenario changes in RBPR2 mice on vitamin A sufficient or vitamin A deficient diets, where we can see a high presence of apoprotein opsins in the retinas of these mice, further confirming that the levels of rhodopsin are quite low in these mice. So to summarize this talk, we can see that wild-type mice can maintain serum and whole body retinoid homeostasis under variable conditions of dietary vitamin A supplementation. RBPR2 mice are unable to maintain serum and whole body retinoid homeostasis. These mice further show decreased total retinoids in the liver, serum, and peripheral tissues, which is most severe under the VAD condition. And finally, in the long term, at 6 months, the RBPR2 mice show decreased ocular retinoids, presence of apoprotein opsin, and decreased visual function. So based on this, our data supports the hypothesis that both genetics and the diet play an important role in whole body vitamin A homeostasis in the support of visual function. So what are some future directions and outstanding questions that we have? So at the moment, we are still figuring out the mechanism for higher serum apo-RBP protein in RBPR2 mice. We think is it due to increased liver or adipose tissue RBP4 mRNA transcription in the disease state, or is it due to reduced serum apo-RBP4 protein clearance? And what is the metabolic fate of unabsorbed retinol-bound RBP4 in RBPR2 mice? Is it redistributed or sequestered into other tissues, or is it excreted? So to answer these first two questions, we are now generating liver and adipose tissue conditional RBPR2 mice. And finally, what is the role of RBPR2 in the intestine? This has not been established as yet. Is it involved in retinol efflux? And for this, we are establishing intestine-specific RBPR2 mice. And a couple of acknowledgements. These are my current lab members, and those highlighted in bold are the ones that did the work, and your former lab members, and then some of my funding sources. And I'm happy to take any questions.
Thank you, Glenn. Let me start as other people will collect their thoughts and to ask you questions. Is there any known mutation in humans related to this receptor?
No, there is not.
Okay. And the second, my question is, it looks like you didn't study yet the cones, right? How they affect it.
Yeah, we have not yet studied cones. We do have some photopic ERGs, but depending upon the light source, if it's red, green, or blue, they're showing different types of pathology. So we're still trying to figure out what's happening in the cones.
Hi, Dr. Lobo. Thanks for the talk. I have a sort of naive question. I wonder how fast is retinol washed out from the body in the settings of normally functioning RBPR4 and RBPR2 and RBPR2 deficient. Is there any data for that?
Yeah.
And we know.
Yeah, so this is the second question that we hope to answer. We are planning to, inject tritium bound retinol to RBP4 so we can follow radioactively what happens to unbound or unabsorbed retinol in these mice. So at the moment, we don't have an answer what happens to unabsorbed retinol because this is a global knockout mouse. And as you saw earlier, that this receptor, this protein is expressed in multiple tissues with the exception of the eye. The mouse model, the global mouse model, cannot be used to answer questions, what happens to unabsorbed retinol? We have to generate a conditional knockout, and for that we're doing a liver-specific knockout, and the liver being the main vitamin A hub. We expect that unabsorbed retinol should be redistributed to other tissues.
Hey, Glenn, this is Philip Kaiser. Nice talk. Thanks for making it so clear. There were two things, two thoughts I had about with your RBP4, increased levels you're observing in the serum. So, normally under vitamin A deficient conditions, you'll have RBP4 actually accumulate in the liver. So I was wondering if you had looked at the livers of those animals to see if you have higher levels there. And then on the other side, with the clearance, have you looked at the urine of those mice to see if you have increased RBP4 in the urine as you would expect if you have an excess of ApoRBP4? Yeah,
very true. So we are currently doing those studies. We have mice in metabolic cages. And to answer your liver question, we have looked at RBP4 in the liver. We do find it high. We're not sure if it's transcriptionally related because, studies have shown different things. It could be related to transcription upregulation, but there are some, concerns about having a consensus what's happening to RBP4 in pathology. As you also know that the adipose tissue can also release RBP4. So we are not really sure if the increased ApoRBP4 is coming from the adipose tissue under pathology. So that's the reason we have to generate adipose and liver conditional mice.
All right, and we have one question from the Zoom, Robert Paul Malchow.
Yeah, hi, thanks. I was curious to know why two separate transporters? What's the advantage of having two separate transporters? Why not just have one?
So that's a good question, right? So the vitamin A, the whole story about vitamin A is very plastic. It's very redundant. So you can, if one system fails, I presume nature has developed a second alternative transport mechanism for vitamin A. You can get it through the diet or also through stored vitamin A. So it could be presumed that, or speculated that vitamin A receptors exist to perform all of the same, they perform the same function. I think it's important if, say, for example, STRA6 is absent from the liver or from placenta, you wouldn't have a human being being born. So I presume that nature has provided us with alternative mechanisms if in case one system fails, the second one is available. We haven't tested that yet, but having a double knockout mouse of STRA6 and RBPR2, but I could speculate that nature has developed systems so that the body does get vitamin A in some form or the other. And it is also regulated as well. So if you have a lot of vitamin A, because you can get hypovitaminosis from consuming a lot of liver, like Eskimos, they would die from hypovitaminosis from consuming polar bear liver. So I presume these receptors regulate how much of vitamin A gets in.
Hi,
Thanks.
Dr. Lobo. My name is Rafał. I'm postdoc in Krzysztof Palczewski lab. A very interesting talk. I have a question. Is there a difference in interaction of STRA6 and RBPR2 with RBP4, like affinity difference? That's kind of what I'm interested in knowing. Thank you very much.
Yeah, so I did not show that study. We did SPR analysis of these two receptors with its ligand. And there are some subtle differences in the binding of RBP4 to either of these receptors. The only downside of that investigation was that we could not get full-length RBPR2 protein in larger quantities, so we had to use small fragments of it with the binding domain. But they seem to have similar function. The activities are a little bit different, but they seem to have similar functions in binding to exogenous RBP4 to internalize retinol into cells. They have different AD values here.
In relation to that, since they're functionally redundant, would expressing STAR6 in the liver rescue the phenotype? And also considering that vitamin A is important in other aspects outside of vision, do you also see defects in immunity in the mice as well?
Yeah. So what we have seen so far is that, and this is unpublished data as well, I can tell you that these mice actually start developing, they start getting glucose intolerant, and they also start getting obese. So we haven't fully teased out that mechanism. Obviously, it's related to spilling retinoic acid. We haven't gone down that route yet, but we have planned some experiments. We just visually can see that the mice are becoming obese, and they do develop a fatty liver, which is not alcohol related. And then coming to your question of overexpressing STRA6 in the liver, we haven't thought of that yet, but that seems to be an interesting way to see if STRA6 overexpression in liver, maybe in vitro, would rescue the RBPR2 phenotypes.
Hi, Glenn. Nice to meet you again. Maybe I just missed the important information about your talk. I am just wondering, is it possible for this RBP receptor 2 have is substrate binding of retinoids? Did it require of RBP4, and did it has other substrates like retinoic acid, or also about people previous questions? I'm just curious about compare with STRA6. It's possible the configuration of retinoids can affect the uptake rate compared with drug 6.
Could you just repeat your second question?
I just thinking about let's for example, maybe for this receptor compare with drug 6, like other configuration like 13 cis, 11 cis retinol, maybe their uptake are different.
Yeah, so the first question is, does RBPR2 have other ligands? So
yes.
At the moment, we believe that RBP4 or retinol-bound RBP4 is the only known ligand. We haven't tested others independently, but the expression of RBPR2 in the intestine it's at the basolateral surface, both in mice. We have preliminary data to show it's at the basolateral surface of the anthrocytes. It could be involved in the efflux, like STRA6 has been shown. So that could be one direct possibility. It doesn't need a ligand, it just directly effluxes retinol. And we haven't tested if there are other vitamin A metabolites that could bind directly to RBPR2. But yes, we do have some preliminary data that shows that RBPR2 under different vitamin A conditions, VAS and VAD, in mice in vivo, its expression can be regulated. So we are trying to figure out, is it based on the promoter of RBPR2 that contains these retinoic acid response elements? So yeah, so one of its direct ligand that acts via its promoter could be all trans retinoic acid. We cannot rule out 9-cis, but at the moment we believe it's all trans. So because it has both RARs and RXRs on its promoter.
Great talk. I have a question about the earlier photos for the babies with no eyes or small eyes. Were they having like a deficiency in the receptor or deficiency in the vitamin A uptake?
Yeah, so that was for STRA6. It was published in 2007. So there's also some variable phenotypes that can happen in those patients. Some patients with STRA6 mutations, if I remember correctly, and Dr. Palczewski can correct me on this, they do have variable phenotypes depending on the vitamin A status of the mother. So it could be that if the vitamin A status of the mother is sufficient or given high doses of vitamin A, the eye phenotypes could be not as severe. So as I said before, both genetics and the diet play important roles in maintaining ocular or retinoid homeostasis. So yes, does it answer your question?
Good morning, Dr. Lobo. Thank you so much for your very interesting talk. I'm Maria Hołowicz from Dr. Palczewski Lab, and I'm wondering, did you observe any difference in the eye morphology between wild type and RBPR2 knockout mice? Thank you.
Yeah, so again, a very interesting question. So we generated knockout mouse model for zebrafish when I was a research scientist or project scientist in Dr. Perkins' lab at the Kola Institute. And that zebrafish model showed severe, visual architecture phenotypes. In the mouse model, we only observe shorter photoreceptor outer segments when the mice are on a vitamin A deficient diet, as compared to these knockout mice on the sufficient diet, which again brings us to the point that the dietary component of vitamin A can rescue some of these phenotypes, as also shown in STRA6 mice as well. So the phenotype is reduced when these the ocular phenotypes are reduced when the RBPR2 mice are maintained on vitamin A sufficient diets, that we can clearly see from these, you can see RBPR2 mice on vitamin A sufficient diets, they don't show as much of rhodopsin mislocalization or apoprotein opsin because they have been fed with a vitamin A sufficient diet. So there is an alternate pathway to get into the eye just through the chylomicrons, and that could rescue some of the visual and ocular pathologies we see in these mice if the diet is sufficient. But when the diet is deficient in vitamin A, they have no way to acquire, store, or distribute vitamin A, these mice.
Hello, thank you for the talk. You mentioned RBP4 and RBPR2 and STRA6 have similar extracellular residues to bind retinol. For the residues that you altered, did they affect the binding of the polyene chain, beta ionic ring, or the hydroxyl group?
So we didn't test that. We just tested exogenous when we applied retinol bound to RBP4. We only tested the physical binding in SPR analysis and in vitro. So we are really not sure what element was being disrupted. But the take-home message from that in vitro study was mutations of any of those proposed binding residues can lead to defective RBP4 binding and retinol intake into cells. So that's a good question to follow up.
Okay, and we have two more questions from the Zoom audience. Michael Redmond.
Hi, Glenn, great talk. I was interested in asking, as you know, RBP4 is required for fetal development, and STRA6 is expressed on the placenta to get that vitamin A across to the fetus. Is RBPR2 expressed in placenta?
Yeah, so that's a really interesting question to ask. So our initial analysis, when we looked at mouse placenta, we did find by qRT-PCR and semi-quantitative, we did find some expression. But since the homology between these two receptors is about 23%, we were not really sure of its biological role in the placenta. We believe that RBPR2 is more related to when, at least in the mouse, is past its P21 stage, and then into maintaining more of a physiological role rather than a development role. Because we do find these, we find litters, and if it had a role in crossing retinol from the placenta into the fetus, I presume we would have abnormal fetuses. We don't see that. So based on our limited experimental approaches, we don't believe that RBPR2 has a role in development as compared to a physiological role in the redistribution of retinol to these other peripheral tissues.
Right. Okay. Sanjay Asrani, are you there to ask your question?
Yes. Hi, can you hear me?
I can.
Wonderful. Hi, Glenn. Thank you very much for the wonderful talk. Have you, by chance, noticed any loss of nerve fiber layer and ganglion cells in these mice that are deficient in the transporters? And the reason I'm asking is that as a clinician scientist, I see this loss of this layer on OCT in our vitamin A deficient patients. And a study in rats had shown some effective protection by all-trans-retinoic acid in the functioning of the ganglion cells. So that's why I'm just curious if you happen to notice that.
Yeah, we haven't done that deep inner retina analysis, but I do know that there are a few papers that talk about the loss of retinoic acid, or when you put animals on a VAD diet, they have defective muscular movement of the eyes. So that could be one area that, would be interesting to look at, but we haven't done a deep dive into inner retina layers here.
If you get a chance, it would be lovely if you can take a look at that.
I would, yeah, sure.
Thank you.
Hi, good. Again, this is Ray Chen from UCI. Wonderful talk. So I have two sort of unrelated questions. So the first question is I'm struck by the complementary expression pattern between STRAT6 and RBPR2. So I'm wondering whether that's related to their function in a sense that whether STRAT6 have a different affinity or efficiency to import, right, the retinoid, and whether the eye, for example, have higher concentration and liver or low. Is that correlation there? My second question is just, RPPR2, based on the talk, is important for storage, the retinoid, and, homeostasis. I'm wondering how the liver sense the concentration and make it release back. I mean, do you can elaborate a bit on that? Thank you.
So your second question is a $1 million question. How does the liver sense when the body requires vitamin A? When the diet is low, or there's some other pathological. That's a $1 million question. We have started to answer that question, and we believe it's like a feedback mechanism coming through retinoic acid. Our preliminary data does show that RBPR2 could be a sensor. So of this decreased whole body retinoid when the intake of vitamin A is low. So as you can imagine, in the fasting state, as we are fasting right now, we're not eating anything. There is no vitamin A intake coming from the diet. So the stored retinal esters in the liver is converted to retinol and then binds to RBP4, and that's a major transport route. So what initiates that cascade? We believe it's the dipping of the level of retinoic acid in the liver that initiates this cascade. We don't have the answers right now, but we do believe that RBPR2 plays a role in this liver to eye nexus or axis. And your second question, we haven't tested what are the requirements for RBPR2 and STRA6 in these different organs? Do they take up retinol from its protein-bound form effectively the same, function or intensity? That would be hard to answer in vivo, I feel. But I feel that they are both separate receptors having similar functions. One in the liver to reabsorb circulatory retinol from RBP4 and into peripheral tissues for storage in times of vitamin D deficiency. And obviously STRA6 in the eye is required to maintain ocular retinoid homeostasis for visual function and phototransduction.
Hello. I just wonder how the two models, RBP2 and STRA6, are comparable or not in terms of the rate of accumulation of unligated rhodopsin, like over age.
Related
is the free opsin accumulates slower in RBP2 model compared to STRA6 model?
Can you just repeat that last part of your question?
So what's the rate of accumulation of unligated free opsin in RBP2 model compared to STRA6 model?
Yeah, so we haven't done that analysis. What is the rate of accumulation of free opsins? But we do find that it is not imaged in the early life of these animals, but it happens in later life. And we presume that these mice, since they are the RBPR2 knockout mice, since they are subjected to vitamin A deficient diets, the vitamin A status in these mice, the whole body vitamin A status in these mice are really low. And it's only in the long term, at the 6 months time point, do we see apoprotein opsins in the eye. We haven't done a time-based or monthly or weekly time-based assays to see how much and how long or what's the initiating time point, because we would need a lot of mice for those to do the sort of longitudinal weekly studies. But our endpoint analysis does show that RBPR2 knockout mice on the VAD diets not only acclimate, not only have apoprotein opsins, but they also acclimate mislocalized rhodopsins, at least at the 6-month time point. We haven't done that at the earlier time points. Yeah.
All right. Thank you, Glenn, so much. Let me finish with 2 more questions, because I'm sure the room just expect those questions from me. So let me ask you, did you look on the structure of the R2? Did you predict the structure, how it's differ? And my last question is, did you look on phylogenetic tree, how this gene is preserved among many different species, whether correlate? And number 3, you're making a conditional knockout in liver. And of course, liver has multiple cells. So how you deal with that when you make a knockout? Is hepatocytes and stellar cells or other cells?
Yeah, good question. So with the structure, we have started to express proteins. We do have properly folded proteins. We're trying to use crystallography or AFM to figure it out. And I think I did contact Dr. Kaiser a couple of years ago to help us with this. And I'm hoping that he would help us with this structural analysis. It does seem that the STRA6, at least from computer modeling, it does seem that STRA6 and RBPR2 are pretty much structurally similar for its function in binding to RBP4 retinol. But that's only computer analysis. So we do have protein at the moment, and we are trying to solve that using AFM or crystallography. And the second question was, how well is this conserved? So I showed you earlier that when you align the protein sequences from zebrafish, mouse, or STRA6 and RBPR2, it seems to be about 21 to 23% conserved. In humans, the STRA6 is expressed on a single chromosome. It looks like RBPR2 is split into 2, and it's expressed on 2 different chromosomes. So how that functions in a human setting is not currently understood. But the mouse model seems to have a single gene, a full gene expressed on 1 chromosome for RBPR2. And you had a third question. What is the third question?
Third question related to liver heterogeneity of cells.
Yeah, so that's a good one as well. So at the moment, we have these albumin gray mice that are crossed with our flox RBPR2. And we do have experiments we've done that seems to suggest that RBPR2 is at least lost, the expression is lost in the liver. To answer your question in depth, yeah, we are currently trying to fractionate between stellate and hepatocytes. And we also have another mouse that we have, a TD tomato mouse, that we can see expression as well. So we have a few different approaches to confirm that the liver is devoid of RBPR2.
All right. Thank you very much. And until next week.
Thank you.